in the stratified epithelium is the transitional epithelium. Transitional epithelium. It is present in the urinary tract, pelvis of the kidney, ureter, and the urinary bladder. This epithelium can change its thickness according to the state of the distension of the bladder. We will study this epithelium when the bladder is empty and when the bladder is full. When the bladder is empty, the thickness of the epithelium ranges from 6 to 8 layers. The basement membrane is non-clear, non-wavy basement membrane. The basal cells are low columnar cells with basal and oval nuclei. The intermediate cells are polyhedral cells with rounded nuclei like the stratified squamous. The top layer is dome shape like that. It has upper convex surface and lower concave surface. It has a rounded nuclei and may be binucleated, has 2 nuclei like that. The surface, the top layer is covered by mucus to protect this epithelium from the harmful effect of the urine. The underlying cells are flask in shape. They are lying in the concavity of the upper one. What is about the full bladder? The full bladder is formed of 2 layers. The basal layer is cubical and the upper layer is flat. This is due to the cells glide over each other by the help of mucus. This is the empty bladder. It has many cells, but the full bladder have 2 layers only, basal cuboidal and the upper is flat. The stratified columnar epithelium, as we it is formed of about 20 layers of cells. The cells are arranged: basal columnar cells, middle polyhedral cells, and top layer is columnar in shape. The stratified columnar, we have 2 types. Non-ciliated, it lines the large ducts of the glands. Ciliated, it lines the fetal esophagus and it is rare. Now we will discuss the glandular epithelium. It is an epithelium which is modified to produce secretion. Classification of the glandular epithelium according to the duct, according to the secretory part, according to nature of secretion, and according to mode of secretion. According to presence or absence of a duct, we have exocrine possess ducts like the salivary glands. Endocrine have no ducts like thyroid, pituitary, and adrenal glands. Mixed glands have 2 types, exocrine and endocrine, like pancreas and liver. The second classification, according to branching of the duct, we have simple duct, 1 unbranched duct. It is present in the intestinal and gastric glands. The second type is compound gland. Have branched duct system. It is present in the salivary glands. The third classification is according to the secretory part. The secretory part may have many cells or one cell. If the number of cells is 1, so we call it unicellular gland. It is present only in the goblet cells in our body. If it is formed of many cells, we call it multicellular gland, and it is formed of many cells. It is present in the salivary glands, pancreas, and liver. According to the shape of the secretory part, may be tubule, alveoli, or tubulo-alveolar gland. The tubule, like intestinal gland, the alveolus may be as the sebaceous and mammary gland, tubulo-alveolar present in the salivary gland and pancreas. According to branching of the secretory parts, they may be non-branched or branched. If it is non-branched, it is present in the intestinal gland. If it is branched, it is present in the salivary gland, liver, and pancreas. The fourth classification is according to the nature of secretion. The nature of secretion may be mucus. It produces a viscid mucus poor in enzyme, for example, the goblet cells and minor salivary glands. Serous glands produce watery solution rich in enzymes like parotid and pancreas. Mucoserous produce both types of secretion, for example, submandibular and sublingual glands. Oily glands secrete fatty secretion like sebaceous gland of the skin and tarsal glands of the eye. Waxy gland secrete waxy secretion, for example, ceruminous glands of the external ear. Cellular glands secrete cells like testes and ovary. According to mode of secretion or method of secretion, we have merocrine glands. These merocrine glands, secretory cells are not destroyed during secretion. For example, salivary gland. Apocrine glands, the apical part of the cells is destroyed during secretion. This is like the mammary gland. Holocrine glands, the whole cells is destroyed during secretion. For example, the sebaceous glands. Now we will study the neuroepithelium. This is a type of epithelium that is modified for receiving of sensory stimuli. The structure of the neuroepithelium, it is formed of sensory cells, tall cells with hairlets on their free surface. This is the sensory cells, has hair cells on its free surface. Their bases are surrounded by free nerve endings. This is the stimulus, will stimulate the nerve fiber. The second cell is the supporting cells. They support the sensory ones. The third cell is the basal cell. It is small pyramidal cells lying on the basement membrane. They act as a precursors for the other two types, or a stem cell for the other two. The sites of the neuroepithelium present in the taste buds, in the tongue, olfactory epithelium in the roof of the nose, organ of Corti in the inner ear. The intercellular junction or cell connections. The second type is the adherens junction. It consists of transmembrane protein bind to similar protein on the adjacent cell in the zonula adherens and desmosome, or to the extracellular matrix in the hemidesmosomes. The third type is the gap junction. This gap junction contains numerous transmembrane channels. These channels allow the passage of inorganic ions and other small molecules from cytoplasm of one cell to the other cell. Thank you.